friends, welcome back to my channel, and today in this video we are going to study about recessive epistasis. But before that, let's understand what do we mean by the term epistasis. Epistasis, it is a type of non-allelic interactions, correct? So gene-gene interaction, it is a type of gene-gene interaction where one gene is interacting with another gene. It is not like interaction between one allele with the another, in which we studied about codominance, incomplete dominance, and dominant recessive relationship. That was for allelic interactions. Now we are studying gene-gene interaction, which is a type of non-allelic interactions where one gene will interact with the another gene present at some another location. Now we are having two genes, gene A and gene B. If gene A is affecting the phenotype of gene B, whether positively or negatively, in that situation, gene A we will call as a epistatic gene and gene B we will call as a hypostatic gene because it is getting affected by the expression of gene A. That means the expression of gene B is affected by the expression of gene A. Because gene A is affecting B, so A is epistatic, B is getting affected, so B is hypostatic. Now, when we study epistasis, we will see that the ratio, which is 9:3:1, the ratio of law of independent assortment, and the test cross ratio, which is always 1:1. Here, because of we are taking two characters, so it will be 1:1:1:1. 4 times 1. When we study monohybrid cross, in that the test cross ratio comes 1:1. Clear? So this two ratio get changed in case of epistasis. Now, first we will study today, that is recessive epistasis, which is also known as supplementary genes. Now, from the word recessive epistasis, so that means that when the epistatic gene, what is epistatic gene? Who will affect other gene? The gene that will affect other gene. So when that epistatic gene is in recessive state, that when it affects the other gene. It is a recessive epistatic. That means the epistatic gene. That means the gene that will affect the other gene. When this epistatic gene are present in recessive condition, that when it influence the expression of another gene B. Clear? That is known as recessive epistasis. So the phenotype get influenced only when epistatic gene is recessive, regardless of the state of hypostatic gene. Now, suppose capital A small a, capital B small b of F1 generation are crossed. In the F2 generation, if it is a law of independent assortment, if it is working as law of independent assortment, what we will get? The ratio is 9:3:1. It is easy to remember the ratio of law of independent assortment. Only remember that ratio, that is 9:3:1. No need to remember or mug up different type of epistatic relationship ratio. What we will remember? Only the law of independent assortment ratio. With this ratio only, we can easily get the recessive epistatic, or when we will study in future the other epistatic ratios. Clear? So it is easy to remember the ratio of law of independent assortment, that is 9:3:1. And also, right from the video of law of independent assortment, I'm telling you to remember this 9. How we get 9? When both the characters are dominant. Right? So when both the characters are dominant, that means from both the characters, one allele should be present as dominant. So here capital A and capital B. The other, the dash one, can be a small, can be a capital, can be a recessive, can be a dominant, doesn't matter. But one allele should be dominant for the expression of the phenotype. So 9 is for the dominant phenotype. So when the dominant phenotype will come? When at least one allele dominant is present. So capital A dash, capital B dash is for 9. This middle two, one dominant, one recessive. So if A is recessive, small a, then the character B should be dominant. For that, one allele should be dominant present. The vice versa for this 3, capital A for the expression of this character and B small b for the recessive one. And all recessive for this 1. So 9:3:1. 9 for the dominant, 1 for the recessive, and the between 3 for the one character dominant, another character recessive. At least this much is easy to remember. Now, in recessive epistasis, what we are saying? A is our epistatic gene. So when A is present in recessive condition, it will affect the phenotype of the B. So where you can see A recessive, this second one and this fourth one. So this second one and this fourth one, you can find that A is in recessive condition. So this two will affect this particular phenotypic ratio part. So now the new ratio will be, the 9 will remain 9, this 3 will change and this 1 will change. So together 3 plus 1, 4. Because in both the situation, epistatic gene is in recessive condition. So both will affect the phenotype in the same way. So in the new phenotypic ratio, because these two will affect the phenotype in the same way, we will add these two. So 3 plus 1, 4. This middle 3, this will again remain the same because here the epistatic gene is in dominant condition. So the new ratio is 9:3:4 or 9:4:3. So it is easy to remember now what is the ratio of recessive epistasis from law of independent assortment. How it affects the test cross ratio. In the test cross, we get 1:1:1:1. This is the ratio. How it will affect? You tell me. This will remain the same. This will remain the same. Here, epistatic gene is in recessive condition. Here, epistatic gene is in recessive condition. So we will add these two. The new ratio will become 1:1:2. So this is recessive epistasis. For this also and for future all the types of epistasis, only remember the law of independent assortment ratio, that is 9:3:3:1. And also along with that, the 9 is for what type of character, 3 is for what type of character, 1 is for what type of character. By that only you can get the ratio for any kind of epistasis. So this is recessive epistasis. One example of recessive epistasis is Labrador Retriever dogs. Their fur color can be black, brown and yellow. Now here epistatic gene A, hypostatic gene B. So according to law of independent assortment, we can get capital A dash, capital B dash. Second one, this is recessive. Third one, B is recessive. Fourth one, all recessive. Correct? So where it will affect the phenotype? This will affect the phenotype and this will affect the phenotype. So this is 9. So 9 is black. This is brown. The 3, where hypostatic gene is in recessive condition. These two, small a, is affecting the phenotype. So here you can see both are yellow. So this is 9, this one is 3, and this both yellow together is 4. So 9:3:4, which we can observe in Labrador Retriever dogs. Now, why it happens? The reason behind this is gene A is actually responsible for deposition of color, either black or brown. When gene A is non-functional, recessive means gene A is now non-functional, both are recessive state. In that, it cannot deposit any color. So it becomes yellow. Now, one question from my end for you all. A pure homozygous black Labrador Retriever is crossed with homozygous yellow Labrador Retriever. In the F2 generation, 150 puppies are opted. Calculate the number of brown puppies in the F2 generation. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.